our team BHSF. I'm Annika.
I'm Reina.
I'm Stephen.
I'm Michael.
I'm Kevin.
In this video, we will introduce you our project, Khronos, the Timer Yeast.
We're going to introduce our project as follows. Baking at home is becoming increasingly popular nowadays, especially during the pandemic when we try to avoid unnecessary crowding. However, many home bakers are troubled with the problem of over-fermentation, which means not being able to stop the fermentation of dough on time, leading to much frustration. This year, we aim to solve the problem by modifying the genes of baking yeast. After rounds of human practices, we refined our design, which mainly composes of a toggle switch and auxotroph yeast.
When we bake at home, fermentation is always a difficult step in the process. The most unmanageable factor which influences the degree of fermentation is the time. Annika, our team leader and a baking lover herself, experienced the problem of over-fermentation when baking. And when talking with our friends who also bake at home, we found that they also struggle from the bad taste and flavor from over-fermentation. The bread over-fermented and that insufficiently fermented can both have unpleasant taste and flavor. It is a tough challenge, especially for the beginners. Therefore, we decided to invent a yeast that ultimately terminates fermentation at appropriate time, trying to help home bakers with their breads.
After the idea first came to us of inventing a timer yeast, we set out to investigate possible implementations for our project from different perspectives. So, we interviewed the bakers of Palette and Farina, two famous bakeries in Beijing. From the interviews, we learned that professional bakers use time as the measure of fermentation process and that fermentation time varies a lot among different types of bread. Both of the bakers thought our idea to be interesting and unique. They also suggested that apart from home baking, our project could also be of value to frozen dough industries, where dough are fermented and then frozen, ready for baking. So we looked deeper into the industry of frozen dough. We learned that during the process of producing frozen dough, in order to put the yeast into dormancy after fermentation, the dough are frozen to a very low temperature in a short time. However, this process will damage the taste and touch of the bread. This was confirmed by our following investigation with two other bakeries, that customers do have a preference to bread made of unfrozen dough. Therefore, an engineered yeast strain that goes into dormancy without freezing will be of great value to frozen dough industry. From these investigations, we set our goal to engineer a yeast strain that uses time as a measure for fermentation process and goes into dormancy after a certain period of time.
With these ideas in mind, we designed our circuit as follows. We use a toggle switch to turn the yeast from an active state into an inactive state. The toggle switch is made of a pair of mutually repressing promoters, P-lag and P-lambda. These promoters are constructed by adding prokaryotic operators downstream of Tata boxes in eukaryotic promoters, which has proven to be an effective way of constructing inducible eukaryotic promoters. When the yeast is active, the C suppressor protein inhibits expression of genes, that is, genes of lac suppressor protein and the toxic protein. Outside the switch, a galactose promoter controls the expression of another lac suppressor gene. When galactose is added into the system, the lac suppressor gets to express, triggering the toggle switch to be turned to the other state. That is the state where the toxic protein begins to express and accumulate, eventually terminates the activity of the yeast. The use of toggle switch allows us to develop yeasts of different fermentation time by changing promoters in the switch. This will make our yeast fitful for different types of bread.
With the preliminary design of our project, we contacted Professor Jianfen Liang from China Agricultural University and asked for her advice on our project. Professor Liang pointed out that the toxin in our design may cause problem in food safety. Even if it is harmless to human body after baking, the notion of using a toxin in food alone can cause repulsion among the public. In addition, Professor Liang suggested that we find alternatives to trigger the switch, for it requires a consistent accumulation of black eye protein. If we used galactose promoter to fulfill the function, the amount of galactose would be difficult to control, for too little galactose would be soon exhausted, and too much may affect the flavor and quality of the bread.
Following Professor Liang's suggestions, we substituted the toxin part with auxotroph yeast to achieve the same goal of putting the yeast into dormancy, and included an integrase into our circuit to have lac inhibitor constantly accumulated after one single stimulation of galactose. Integrases are enzymes from virus that integrates viral DNA into the genome of its host as specific recognition sites. However, if we put phage and bacterial attachment sites against one another on each side of a DNA sequence, the integrase will recognize the attachment sites and flip the sequence in between. This mechanism has been widely used in many iGEM projects. So here's the improved version of our design. In the beginning, the complement gene for auxotroph is expressed, allowing the yeast to perform its function. When galactose is added into the system, integrase is expressed and inverts the sequence of the promoter upstream of the lac inhibitor. So that lac inhibitor begins to accumulate. Even if galactose is removed afterwards, then after the toggle switch is triggered, the expression of the complement gene stops, leading to a drop in its concentration and putting the yeast into dormancy for lack of the nutrients. When baking with our yeast, the yeast should be activated in a solution containing galactose to initiate the timing machinery before using. And according to our human practices, activation of yeast is a common procedure in baking.
As mentioned before, we want to adjust fermentation time by substituting promoters with different strengths in the toggle switch. But how to correspond promoter strengths to fermentation time? That is what we try to figure out through modeling. We built a model as shown here. It has promoter strengths as independent variable and the concentration time course of two protein as dependent variables. After construction of the whole system, we'll first test the model with one or a few pairs of promoters. Then we'll make some adjustments to our model using the experiment results. After these refinements, we'll be able to pick out proper sets of promoters by calculation according to different needs of fermentation time.
Due to the pandemic, we were not able to carry out our experiments as planned. However, we did learn the very basic experiments of molecular cloning and began to construct our first vectors. Here's the plan for our experiments. Because our circuit is complicated and contains many parts, it would be difficult for us to make refinements if we test the whole system from the beginning. So, we will separate the work and test individual parts before assembling them into the integrated system. To test the validity of the toggle switch, we will first need to test the novel prokaryotic promoters that we designed. Using fluorescent proteins as reporters, we will be able to test whether the promoter can be inhibited by inhibitor proteins. Then, we will integrate two of these promoters with different operators into a toggle switch and test whether it can be triggered. Although the integrase from Mycobacterium phage BXB1 needs no special cofactors to function, we want to test its performance in the yeast before putting it into our system. To this end, we will have the integrase aligned sequence of the fluorescent protein to the promoter and examine the fluorescence to verify the function of the integrase. After all the work above is done, we will integrate the parts into one system and test the function of the whole system.
Although limited in experiment conditions, we obligated ourselves in many other constructive works for our project in synthetic biology. We collaborated with Keystone as well as a couple of other iGEM teams to host a biofair where we introduced our projects and promoted synthetic biology to the public. One of the most commonly heard feedback is the concern for the safety of genetically modified organisms. This made us realize the urgent need for spreading the positive information of genetic engineering. To this end, we designed a series of promotional activities for synthetic biology, aiming for audiences from primary school students to high school students. For high school students, we designed some questions related to these knowledge and did some prize-giving quiz with the student. The students told us that they learned lots of new things about biology and genes from these activities, and that they were looking forward to more activities of this kind. For primary school students, we gave them lectures on some basic concepts and usage of gene engineering and synthetic biology, and we used our project as an example to illustrate our points. Many students were pretty interested in our project and lectures and asked us lots of questions about these topics after the lectures. It was a relief to us how juveniles at and below our age are ready to accept new technologies. It showed us that with joint effort from science communities from all over the world, the public view towards genetic engineering can be changed in the near future. We also posted many videos related to our project and synthetic biology on various social media such as YouTube, Bilibili, and WeChat channels. The total click is over 100,000. Lots of feedback was received from their comments, including some from famous bakers who also shared their experience in baking in return.
In the future, we will further refine our design to meet the need of secondary fermentation for some other kind of bread. The reversibility of toggle switch and usage of auxotroph yeast instead of toxic protein gives possibilities for such design, but more details are yet to be settled.
In the end, we want to give our most sincere thanks to our instructors, our parents, our classmates, our collaborators, and all those who have helped us in any ways. Thanks for watching our presentation. We will continue with our project and retain our love and passion in synthetic biology. Thank you.